bioenergetics related to the process of photosynthesis. We go to question number 1. Question number 1. Non-cyclic electron flow in chloroplasts results in the production of A. ATP only, B. NADP only, C. ATP, NADPH plus O2, D. ATP and NADPH only. When we are dealing with non-cyclic photophosphorylation, in the process of photosynthesis, this deals with light reactions. The light reactions occur in the grana. Non-cyclic photophosphorylation is also termed as a Z scheme. It has been studied by Hill and Bendel. There is the involvement of both the photosystems, PS1 and PS2. As you understand, when you take non-cyclic photophosphorylation, initially the sunlight or your photon of light hits a chlorophyll bed, and when it hits a chlorophyll bed, the chlorophyll molecules get excited. And when the chlorophyll molecules get excited, the chlorophylls, the electrons are funneled into a reaction center. The reaction center can be P680 or P700. From P680 and P700, simultaneously a series of two photochemical reactions start, and both the reactions are connected, and when they are connected we have the light reactions. So among the light reactions in non-cyclic photophosphorylation, the characteristic feature of non-cyclic photophosphorylation is the electron is not returned. The second important feature is photophosphorylation, means the production of ATP. Now, when you take non-cyclic photophosphorylation, if you look at the sort of reactions in non-cyclic photophosphorylation, we have is A, we have is photolysis of water. There is water split. When water is split, there is liberation of hydrogens. Along with the liberation of hydrogens, there is also the liberation of electrons. So initially, photophosphorylation, for the light reactions, we have the photolysis of water. Parallelly to, we have the electron flow. When there is an electron flow, there is the formation of ATP. ATP gets formed between cytochrome B6 and F. Third, the electrons are funneled. We have photosystem 1 on one side and we have photosystem 2 on the other side. So we have the photolysis of water here. Water gets broken down. Then electrons are released. Electrons go through a series of acceptors and they come and fall down into photosystem 2. Once it falls into photosystem 2, from photosystem 2 it enters into ferredoxin. And from ferredoxin there is a formation of NADPH2. Thus, in the case of non-cyclic photophosphorylation where the electrons are not returned, when you take the non-cyclic photophosphorylation, it is just one way. The electron does not have a sort of a return path. There is no return path of the electron. And in the process, there is the formation of ATP between cytochrome B6 and cytochrome 7. There is the formation of NADPH2. ATP and NADPH2 go to form the assimilatory power, and the assimilatory power is used in the dark reactions. Parallel to this, because of the photolysis of water, oxygen is released. Hence, among the choices, the C choice says ATP, NADPH and O2. The right answer is C. We go to question number 2. Tropical plants like sugarcane show high efficiency of carbon dioxide fixation because of A. TCA cycle, B. EMP cycle, EMP pathway, C. Kelvin cycle, D. Hertzsprung pathway. When you take tropical plants, tropical plants include crops which belong to monocots like rice, maize, sorghum, sugarcane, and predominantly they are sun crops, and sun crops are better with relation to photosynthetic efficiency, that is the trapping of the light energy and converting it into sucrose. So when you take tropical plants like sugarcane, which is saccharum, and they have a high efficiency method of photosynthesis which is termed as a C4 pathway. Now when you take the C4 pathway, it is also termed as a Hertzsprung pathway, because it has been discovered by two important scientists whose names are Hertz and Slack. Now why does the C4 pathway, why is it more efficient when compared to the sort of a C3 plants? Number one is it can quench carbon dioxide very efficiently. Whenever carbon dioxide is available in the atmosphere, it can quickly, the plants can quickly suck in carbon dioxide, and thereby carbon dioxide is available. And in these particular plants, another interesting feature is they have a sort of a specialized anatomy called the Kranz anatomy. So in the Kranz anatomy, we have the mesophyll cells. If you take a typical leaf has got an epidermis, then it has got the mesophyll tissue, then it has got the vascular bundles. Surrounding the vascular bundles, we have the sort of a bundle sheath. The bundle sheath cells have got specialized chloroplasts, and the mesophyll cells have got specialized chloroplasts. Thereby, in the case of the C4 pathway, there is dimorphic chloroplasts. Then again, when you take the C4 plants, they have specialized enzymes. Along with the traditional C3 enzyme, which is termed as ribulose diphosphate carboxylase, they also have another enzyme called phosphoenol pyruvate carboxylase. So there is a presence of RUDP case, which is the most abundant protein which is available on planet Earth. In addition to this, they have another specialized enzyme called phosphoenol pyruvate carboxylase. So phosphoenol pyruvate carboxylase it can primarily suck in carbon dioxide, and it does internal carbon dioxide fixation. Thereby, plants become more efficient with relation to trapping the sort of a carbon dioxide. So under conditions when there is high temperature, there is water stress, the C4 plants have spatially separated the photosynthetase. There is spatial separation of photosynthetase. That is the photosynthetates, that is the carbons, the C3 and the C4 and the C5 and the 6,6 compounds get differentiated, and they get segregated between the mesophyll cells as well as the bundle sheath cells. So spatial separation of photosynthetates is a unique feature of C4 plants. And again, when you take the tropical plants, they are very, very efficient because they show diurnal chloroplast, and again they have specialized enzymes like phosphoenol pyruvate carboxylase. So this method of pathway, which has been discovered in tropical crops by Hertz and Slack, is popularly termed as the Hertz-Slack pathway. Otherwise, it's also termed as the dicarboxylic acid cycle. While the choices of TCA cycle, right, it is nothing but the Krebs cycle, then EMP is an alternative pathway, and Kelvin cycle is a traditional cycle of photosynthesis with relation to the path of carbon. So among the choices, the right answer is Hertz-Slack pathway, that is answer is D. We go to question number 3. Photosynthesis is a redox process in which A. Carbon dioxide is reduced and H2O is oxidized, B. NADP is reduced and RUBP is oxidized. C. Carbon dioxide, NADP and H2O are reduced. D. O2 acts as an oxidizing agent and water acts as a reducing agent. When you take the process of photosynthesis, that is the process of formation of complex carbohydrates from simpler compounds like carbon dioxide and water. Carbon dioxide and water in the presence of chlorophyll and sunlight manufactures the basic glucose product, that is C6H12O6, plus there is evolution of oxygen. Now, during photosynthesis, primarily we understand that carbon dioxide gets reduced. The meaning of the term reduction is there is addition of hydrogen. Once there is addition of hydrogen, it goes to form C, H and O, which are the basic units for starch. And similarly, H2O is oxidized. Oxidized means the liberation of oxygen and the separation of the hydrogen and the flow of electrons. So, primarily because of the flow of electrons, whenever in the process of photosynthesis we have an electron transport system, and in the electron transport system we have several chemical compounds, and each of these particular chemical compounds, like say A, B and C, can get simultaneously oxidized and reduced. They can get simultaneously oxidized and reduced. So, photosynthesis is a unique process wherein it is a redox process. It is called a redox process because carbon dioxide combines with hydrogen and the process gets reduced and it results in the formation of CHO. Hence, the right answer is A. Question number 4. The first carbon dioxide acceptor in C4 plants is A. Phosphoenol pyruvate, B. Ribulose 1,5-diphosphate, C. Oxaloacetic acid, D. Phosphoglyceric acid. When you take the C4 pathway, in the C4 pathway, there is the formation of 4 carbon compounds. The most important 4 common compounds which are formed in the C4 pathway, that is pathways of photosynthesis of highly efficient plants which are tropical plants. The 4 carbon compounds are malate as well as aspartate. The malate and aspartate are the 4 carbon compounds which are formed from phosphoenol pyruvate. Phosphoenol pyruvate is a 3 carbon compound. The 3 carbon compound combines with the carbon dioxide. There is 1 carbon from here and it combines with the 3 carbon compound and it results in the formation of phosphoenol pyruvate. So, the initial acceptor of carbon dioxide is phosphoenol pyruvate. Hence, the answer is A. Question number 5. In C4 plants, A granule chloroplasts are found in A. Chloroplasts of guard cells, B. Epidermal cells of green stem, C. Bundle sheath cells, D. Mesophyll cells. Now, if you take the C4 pathway, as you understand, the C4 pathway occurs both in association between the bundle sheath cells and the mesophyll cells. So, if you take the bundle sheaths, if you take a typical leaf will have an epidermis, then it will have the mesophyll tissue, then it has got the vascular bundle. Now, surrounding the vascular bundle, we have the bundle sheath. This is termed as a bundle sheath. So, we have a bundle sheath cell and again we have got mesophyll cells. Now, in the bundle sheath cells, when you look at the sort of a chloroplast, we are dealing with chloroplasts in the bundle sheath cells of a C4 plant. If we take the chloroplasts are agranal. The meaning of the term agranal is grana is absent. So, because grana is absent, light reactions cannot occur in this sort of a agranal in these sort of a chloroplasts. So, photosynthesis is a sort of a short circuit process, or it is a more efficiency process wherein directly the carbon dioxide is caught by an efficient enzyme called phosphenol pyruvate carboxylase, and directly the load goes into the sort of a dark reactions. So, instead of going through the entire process of the light reactions, quickly dark reactions get in, and once the dark reactions come in, we have the generation of more of ATP. So, once there is more of ATP, there is more formation of starch. So, high starch content, more of photosynthesis, and more of dark reactions. And thereby, if you take the bundle sheath cells, in the bundle sheath cells, we have the presence of starch grains, which are larger. So, presence of larger starch grains is attributed to the occurrence of dark reactions as well as high formation of starch, and again it is because of agranal situation. Hence, for question number 5, the answer is C. Question number 6. Photosynthesis is A. Light trapping process, B. Energy stabilizing process, C. Energy yielding process, and D. All the above. When you take the most important process which occurs in the biosphere, which is termed as photosynthesis, green plants including algae and a few photosynthetic bacteria can primarily trap solar energy. Now, solar energy comes in the form of a photon of light. And when you take the photon of light, and when you exactly want how much of the light energy and how much of oxygen is liberated in the light energy, we are dealing with relation to quantum. So, quantum or photon trapping is primarily termed as a light trapping process. So, the light trapping process relates to the light reactions. During the light reactions of photosynthesis, there is the formation of, that is, the trapping of solar energy occurs on a chlorophyll bed, and all the chlorophyll molecules funnel the electrons to a light harvesting center. And from the light harvesting center, electrons again go through a series of chemical compounds which are termed as chemical compounds which have oxidation reduction states. So, depending upon the redox potential, the electrons are transferred, thereby adenosine diphosphate can get converted to adenosine triphosphate, as well as NAD, that is nicotine adenine dinucleotide, can get converted to NADP. This entire process which occurs in the grana and within the sort of a thylakoids is termed as a light trapping process, which is part 1 of photosynthesis. Then B, we have is energy stabilizing process. That is, the ATP and the NADPH2 are primarily used as a dark reaction, and these dark reaction stable products is termed as a assimilatory power, is something like a sort of a battery which you give it to the sort of a chloroplast. So, when you have the NADP as well as when you have the ATP of the dark reactions, biochemical reactions start. They utilize the ATP and the NADPH2, and thereby carbon dioxide gets fixed by means of a most important enzyme which is termed as ribulose diphosphate carboxylase. So, in the presence of RUDP carboxylase and carbon dioxide, there is a formation of phosphoglyceraldehyde and glyceraldehyde phosphate, and thereby we have the synthesis of C3 as well as C4 and C5 sugars, which is primarily termed as an energy stabilizing process. When we come again back to photosynthesis, in addition to light trapping and energy stabilizing, it is also an energy yielding process. The reason why we say there is an energy yielding process is, during photosynthesis, solar energy gets converted to biochemical energy, and the biochemical energy is synthesized and it is stored in the last or the terminal phosphate of the adenosine triphosphate. Hence, the photosynthesis includes light trapping, energy stabilizing and energy yielding process, and the right answer is all the above, that is, the answer is D. Question number 7. in CAM, that is cross solution acid metabolism? A. Both C3 and C4 cycles are present in the mesophyll cells only. B. Both C3 and C4 cycles are present in the bundle sheath cells only. C. There is no C4 pathway cycle. D. C4 occurs in the bundle sheath and C3 in the mesophyll cells. A look at CAM. CAM is also termed as cross solution acid metabolism. This is a specialized type of metabolism which occurs in succulent plants. Like if you take pineapple, or you take spinach, or you take desert plants which store lot of water like cactus. So in these particular plants wherein which exists in deserts where the temperature is extremely high, there is a stomatal closing, and they have a specialized type of mechanism which is termed as cross solution acid metabolism, which is otherwise termed as LOAM, leaf organic acid metabolism. So in CAM plants, primarily the stomata are closed during the night. The stomata are closed during the day. Because the stomata are closed during the day, there is no gaseous exchange. Thereby, during the day, there is acidification. That is, deacidification or accumulation of acids occurs during the day. So during the day, the stomata are closed. Then there is internal acidification, that is, acids get stored in the vacuoles. And primarily here in this process, we have both the C3 and the C4 cycles. During the day, primarily the C4 cycle operates. There is a presence of phosphoenol pyruvate, and phosphoenol pyruvate catches a little bit of the carbon dioxide and it gets converted to malic acid and aspartic acid. So the malic and the aspartic acid get stored in the vacuoles, and the vacuoles are present in the succulent tissues during the day. Now during the night, the stomata are open. There is deacidification, that is, the malic acid and the aspartic acid move from the vacuoles, and when they move from the vacuoles, they decarboxylize, that is, they liberate internal carbon dioxide. The internal carbon dioxide in the cross solution acid metabolism is taken up and it's accepted by ribulose diphosphate carboxylase, and photosynthesis occurs. So primarily there is ATP generation during the sort of a day internally when the stomata are closed. So here we have the operation of both the C3 and the C4 cycles, especially in a common tissue which is termed as the sort of a leaf mesophyll tissues. So there is no presence of a bundle sheath cell. All leaf mesophyll tissues carry both C3 as well as C4 photosynthesis, and it's a specialized sort of photosynthesis to avoid stress. Hence the answer is A. Question number 8. The evolution of oxygen during the process of photosynthesis is from A. water, B. carbon dioxide, C. oxygen from the atmosphere, D. water and carbon dioxide. The question asks, during the process of photosynthesis, what is the source of oxygen? So we already know that, I already understand that when you take oxygen, oxygen is evolved primarily from water. This is seen in the sort of a bubble experiment. That is, if you take hydrilla, a plant, an aquatic plant, and you put it in a beaker of water, and if a thistle funnel is put on the plant, there is evolution of oxygen bubbles. So this is the first evidence to state that oxygen is evolved from an aquatic plant, and the source of oxygen is from water. The second method of how exactly you know that the source of that is oxygen comes from water is when you take photolysis of water and you are using certain other sort of a chemicals like ferricyanide. The ferricyanide can immediately take in the sort of a hydrogen, and exactly there is the liberation of oxygen. So these are primarily termed as the sort of a photolysis of water, they are termed as reducers. There are several sort of a chemicals which also reduce, and there is evolution of oxygen. So these two experiments primarily determine that the evolution of oxygen during photosynthesis is from water, and the answer is A. Question number 9. The synthesis of one molecule of glucose during Kelvin cycle requires A. 18 molecules of ADP plus 12 molecules of NADPH2. B choice, 12 molecules of each of ATP and NADPH2. C. 12 molecules of ATP and 18 molecules of NADPH2. D. 6 molecules of ADP and 12 molecules of NADPH2. We understand that when you take Kelvin cycle, during Kelvin cycle there is the need for the synthesis of glucose, that is C6H12O6. It means there should be 6 times the movement of carbon in the cycle. We have 6 rounds of movement of the carbon. Now for every movement of carbon, every cycle, for one single cycle, one cycle of carbon which goes through Kelvin cycle, we primarily have 18 divided by 6, that is we have 2 ADP molecules getting generated. And similarly for every cycle we have 12 molecules of ADP, 12 molecules of ADP getting formed, 12 molecules of NADPH2 getting formed. So primarily we have is 18 ATP molecules and 12 molecules of NADPH, that is per cycle we have the formation, there is a need for or the liberation of 2 NADPH. So 2 into 6 is equal to 12. Thereby they can primarily ask you per carbon what is the sort of a generation. You have 2 ADP molecules as well as again you got primarily 2 NADPH2 molecules. Hence for 6 cycles we have got 18 ADP molecules of ADP as well as 12 molecules of NADPH2. Hence the answer is A. Question number 10. Red drop phenomenon can affect and influence one of the following: A. Chlorophyll, B. PS1, C. The accessory pigments, D. PS2. When you take red drop, red drop means the decrease in the photosynthetic efficiency whenever the spectrum is on the lower end. So primarily when you take photosynthetic reactions, we have photosystem 1 and again we have got photosystem 2. So when you take photosystem 2, it relates to P680, and when you take photosystem 1, it relates to P700. So this is the lower end of the photosystem 2. So whenever we take plants or we take microorganisms, we take algae, and when you give them red light, we find that the photosynthetic efficiency stumps or it falls or it declines. So this primarily influences the lower end of the spectrum, that is P680. So because it influences P680, it primarily affects photosystem 2 or pigment system 2. Hence red drop decreases and it can affect the photosystem 2 or the P680 system, and the answer is D. Question number 11. The rate of photosynthesis is doubled for every dash rise of temperature until the optimum is reached. Choices are A 20 degrees centigrade, B 18 to 35 degrees centigrade, C 75 degrees centigrade, D 10 degrees centigrade. Whenever we take photosynthesis, photosynthetic rate, the rate of photosynthesis is always doubled, that is 2x, for every 10 degree rise of temperature. For every 10 degree rise, the photosynthetic rate is doubled. Maybe this is one of the reasons why if you take tropical plants or if you take plants which have got higher photosynthetic efficiency like C4, they show higher photosynthetic efficiency until the optimum is reached. The optimum performance of photosynthesis is normally between 30 to 35 degrees centigrade. So that's one of the reasons why if you take tropical plants are more productive than temperate plants. And the tropical regions are rich with relation to biological diversity. So photosynthetic rate always gets doubled whenever there is an increase in 10 degrees centigrade. Hence, the answer is D. Question number 12. In non-cyclic photophosphorylation, photosystem 1 is reduced by A. Electrons from photosystem 2, B. Hydrogen from photosystem 2, C. Electron from ferredoxin, D. Hydrogen from water. When you take non-cyclic photophosphorylation, when we are dealing with non-cyclic photophosphorylation, it means we are dealing with a Z scheme. And in non-cyclic photophosphorylation, if we have photosystem 1 and photosystem 2, electrons move via photosystem 2, they go to a compound called A, then they go through a series of compounds called B, C and D, they enter into the photosystem 1, and again from here it goes to a compound called E, and later on it goes through this particular pathway. So in non-cyclic, the electrons are not returned. So photosystem 1 is reduced. Reduced means it takes up an electron. When it takes up an electron and gives an electron, the electron source is primarily from photosystem 2. So photosystem 2 generates a series of electrons. These electrons go and they'll fall into the sort of a funnel which is called photosystem 1. So thereby here photosystem 1 is reduced by electrons from photosystem 2. Hence the answer is A. Question number 13. Post elimination outburst of carbon dioxide means A. Photosynthesis, B. Photorespiration, C. Hatch-Slack pathway, D. Dark reactions. In crops, when we take photosynthesis, along with photosynthesis, there is also light induced respiration. That is parallel to photosynthesis, there is also light induced respiration. So during light induced respiration, in the presence of light, any sort of an oxygen which is available outside the plant is accepted by the plant and it is taken by the enzyme ribulose diphosphate. Normally we understand if you take ribulose diphosphate can exist as dual roles. It can go to form ribulose diphosphate carboxylase which can catch up carbon dioxide. Otherwise, here we have got ribulose diphosphate oxygenase. So ribulose diphosphate oxygenase takes in the sort of an oxygen and performs respiration in the presence of light. So whenever there is a flashlight, there is oxygenation, and because of the process of respiration, internally carbon dioxide is generated. There is internal carbon dioxide generation. So the generation of carbon dioxide which is after elimination is termed as post elimination. This is called an outburst. So, a post-elimination outburst of carbon dioxide is primarily attributed to a process which is termed as photorespiration. Now, photorespiration has been studied by a scientist whose name is Decker and Teo. By a group of scientists called Decker and Teo, and it has been studied in tobacco. People believe that photorespiration is a wasteful process. Because it sucks in the sort of an energy so that carbon dioxide is not accepted by ribulose diphosphate carboxylase. But however, photorespiration has a very important role with relation to preventing of free radical damage. It prevents free radical damage. That is, supposing the temperature goes very high. So, the temperature goes very high, it will prevent the chloroplast from getting bleached. So, if there is a bleaching of the chloroplast, the photosynthetic efficiency drops. So, it acts like a sort of a cover process, or it just acts like a sort of an umbrella on the sort of a process of photosynthesis. So, when you take question number 13, post-elimination outburst is primarily related to the process of photorespiration which occurs in green plants. The answer is B. Question number 14. One of the following can efficiently pick up carbon dioxide even at the lowest available concentrations. A. Spirogyra, B. Chlorella, C. Saccharum, D. Helianthus. When you take plants, plants could include algae like Chlorella or even Spirogyra. It may also include a sort of a C3 plant like Helianthus or sunflower, and plants can also include C4 plants. C4 plants can be Saccharum, that is sugarcane. In addition to this, it could also mean maize and barley. Maize, barley, sorghum. Then it can also include CAM plants like pineapple. So, when you take the C4 plants, as you understand, C4 plants have got the special ability of capturing the lowest amount of carbon dioxide whenever it is available around a leaf, even with relation to minimum of carbon dioxide. If it is available, this carbon dioxide is immediately caught by the C4 plants due to the presence of a special enzyme called phosphoenol pyruvate carboxylase. So, they can do efficient trapping of carbon dioxide. So, C4 plants, primarily due to the presence of phosphoenol pyruvate carboxylase, acts like a sponge and quenches up maximum carbon dioxide so that the photosynthetic efficiency of the particular plant keeps on going. Like if a plant is infected by a rust disease, or if it is under water stress, or else a plant has got to immediately put on to flowering as well as go to open the sort of leaves. So, under these three conditions or under stress conditions, plants normally can even shift from a C3 plant to a C4 plant. If a grass plant has got to adapt, it has got to grow very fast and there is limited number of resources. Under limited number of resources, it shifts from a C3 to a C4 pathway. So, C4 plants primarily the advantage is even when there is the availability of carbon dioxide is the lowest, it can photosynthesize. Hence, the answer for this is Saccharum, which is it is sugarcane and it is a C4 plant. Question number 15. Chlorophyll molecule at the core has A. Manganese, B. Iron, Fe, C. Mg, magnesium, D. Copper. When you take a chlorophyll molecule, the most universal molecule is chlorophyll A. Chlorophyll A is termed as a universal molecule. or it is also called as a primary molecule, which is involved in photosynthesis. Any chlorophyll molecule has got a sort of a head and it has a phytol tail. So, the porphyrin head has in its center, it has got the presence of magnesium. Magnesium is the core element which is present in the porphyrin head of the chlorophyll molecule. Hence, among the choices we have primarily because of the presence of magnesium, the answer is C. Question number 16. CAM plants avoid photorespiration by A. Performing Kelvin cycle by night, B. Fixing carbon dioxide into organic acid in night and releasing carbon dioxide in day, C. Using PP carboxylase to fix carbon dioxide to RUDP, D. Keeping the stomata closed during the day. When you take dissolution acid metabolism plants, the prime objective of dissolution acid metabolism is conservation. Hence, they keep the stomata closed during the day. Now, what do the CAM plants do is, they take up carbon dioxide and they do night cycling of carbon dioxide. In night, carbon dioxide is accepted by phosphoenol pyruvate carboxylase and it is taken to form malic acid. As well as along with malic acid, it also results in the formation of aspartic acid. So, the malic acid and the aspartic acid are organic acids which are synthesized and stored in the night in the vacuoles. So, during the night, organic acids assimilate in the leaf tissues, in the leaf mesophyll tissues, and the same carbon dioxide which is trapped during the day, when the stomata are closed, the carbon dioxide is recycled. It is recycled internally. So, internal recycling of carbon dioxide occurs and carbon dioxide is accepted by ribulose diphosphate carboxylase. So, thereby it avoids the process of especially availability, it does not facilitate the process of availability of oxygen to ribulose diphosphate carboxylase. Because if oxygen is available, it will get converted to ribulose diphosphate oxygenase. Whereas, carboxylase is very much active during the day, phosphoenol pyruvate is active during the sort of night, and again that photorespiration is basically avoided in the case of the CAM plants. Hence, among the choices we have B. Fixing carbon dioxide into organic acid in night and release of carbon dioxide in the day has the right choice, and the answer is B. Question number 17. Carbon assimilation during dark reactions was detected by using which of the isotopic forms of carbon dioxide? A we have is 11 CO2, B is 14 CO2, C is 12 CO2 and D is 13 CO2. When we look at the path of carbon, or it is also called a sort of a tracer technique. The tracer technique has been studied by Kelvin, who had been awarded the Nobel Prize for discovery of the dark reactions of photosynthesis. Now, dark reactions are predominantly biochemical reactions. So, in the biochemical reactions, what happens when we have primarily a sort of a 2 carbon or a 3 carbon or a 4 carbon compound, and what really happens in the dark reactions? In the dark reactions, we have a single carbon can combine with another second carbon to form a sort of a triose carbon. A triose carbon can in turn combine with 2 more carbons to form a pentose compound. A pentose compound can combine with a single carbon and form a sort of a hexose sugar. So, if you exactly want to know in a biochemical reaction, we want to exactly know what is the compound which is getting formed, we need to primarily trace it out. So, the trace is predominantly done by using radioisotopic carbon dioxide. The radioisotopic carbon dioxide which is used is 14C. 14C, and the technique is termed as radioautography. By radioautographic technique, by using a radioisotopic 14C, Kelvin and Basham established the path of carbon. Hence, among the choices, the answer is 14C, that is, answer is B. Question number 18. Both photosynthesis and respiration require A. Chloroplasts, B. Cytochromes, C. Mitochondria, D. Sunlight. What exactly is common between the most important reactions in a plant? When you take photosynthesis and respiration, in photosynthesis and respirations, always there are a lot of oxidation reduction reactions. For oxidation reduction reactions in photosynthesis and respiration, which there is the involvement of the electron transport system. An electron transport system in both mitochondria as well as in photosynthesis is predominantly made up of cytochromes and also compounds like plastocyanin and plastoquinone. Now, what exactly are cytochromes? Cytochromes are made up of compounds like cytochrome B6, cytochrome F, and again you got cytochrome C. Now, cytochromes are predominantly compounds which are rich in heme, iron. They are iron complexes which can easily get oxidized and reduced. That is, they can act simultaneously as an electron acceptor as well as an electron donor. So, the compounds which are a part of the sort of an oxidative phosphorylation in the case of mitochondria, in oxidative phosphorylation in the case of mitochondria, it occurs in the F1 particles, terminal oxidation in the electron transport system. Whereas, in photosynthesis, in the light reactions of photosynthesis, we have the electron transport system which links cyclic and non-cyclic photophosphorylation. So, both in photophosphorylation, that is, we have got cyclic and non-cyclic photophosphorylation as well as terminal oxidation, there is the involvement of cytochromes. So, cytochrome is a common point between photosynthesis and respiration. Hence, we have the right answer is cytochromes, that is, answer is B. Question number 19. Synthesis of one molecule of glucose involves dash turns in the Kelvin cycle. A 3, B is 6, C is 12, D is 16. As you primarily understand, when you take the Kelvin cycle, we have got carbon dioxide combining with water in the presence of chlorophyll white, forming a molecule of glucose which is termed as C6H12O12, C6H12O6. So, in order that we get 6 carbons, the carbon which enters into by photosynthesis has got to go through at least 6 turns. So, when it goes through 6 turns of carbon dioxide, that is, dark reaction primarily involves 6 molecules of carbon getting converted to form the sort of a C6. Hence, it involves 6 turns. So, the right answer is you have B, which indicates 6 turns. We go to question number 20. Identify from the following a characteristic pigment associated with chlorophyll B molecules. We have A is plastoquinone, D is ferredoxin, C is cytochrome, D is plastocyanin. When you take the light reactions, or the photochemical reactions in photosynthesis, we understand that we have had photosystem 1 and also photosystem 2. So, if you take photolysis of water and electrons getting into photosystem 2, electrons primarily are accepted by a very important compound called quinone. Now, quinone primarily is made up of even accessory pigments like chlorophyll B. Chlorophyll B is an accessory pigment. They are accessory pigments which are associated with quinone. So, from quinone, the electrons move through electron transport system ETS, of which we have the most important components are the cytochromes. Then it gets funneled into photosystem 1. So, it is a loop line. It comes into photosystem 1. Now, from photosystem 1, it enters through ferredoxin, FD. And en route, it touches even plastocyanin. It touches plastocyanin. So, primarily the first electrons from here, they are accepted by quinone. So, plastoquinone is the right answer. It is a characteristic pigment with chlorophyll B accessories. The answer is A, which relates to plastoquinone. Question number 21. The law of limiting factors was proposed by A. Robin Hill, B. E. Emerson, C. F. F. Blackman, D. is D. Ornan. The law of limiting factors was proposed by F. F. Blackman. So, according to F. F. Blackman, when you take a process like photosynthesis, photosynthesis is a process which is determined by several factors, like say n number of factors. It may be availability of light, light intensity, carbon dioxide, oxygen, chemical compounds or water. It is decided by several factors. Now, if we want to look at the rate of photosynthesis, if we want to calculate the rate of photosynthesis, or we want to improve the efficiency of the rate of photosynthesis, this in turn is determined by the factor which is in slowest supply. It is similar to 10 runners who are just running on a race. The person who wins or the team which wins is made up of components, say players like A, B, C, D, E, or on a race. They are just like the sort of factors. So, if one of the factor is the slowest, this in turn will affect the rate or the efficiency of the run. So, thereby the factor which is present in the slowest, which is supplied at the slowest, will determine the efficiency rate of photosynthesis. This has been studied by Blackman and it is termed as the law of limiting factors. The answer is C. Question number 22. NADPH results during A. photosystem 2, B. photolysis of water, C. evolution of oxygen, D. photosystem 1. Now, when you take the process of photosynthesis, if we have P700, which is called the light harvesting complex, which is otherwise termed as photosystem 1. In photosystem 1, which is on the right, we have the electrons falling from the electron transport system. Once they fall into P700 reaction center, it is also called the reaction center, electrons in turn are funneled into ferredoxin. So, once they are funneled into ferredoxin, from ferredoxin they are captured by NADP and it is converted to NADPH, which is called the assimilatory power. Thereby, NADPH results during the process of photosystem 1, which is one separate sort of a component of photosynthesis. So, it is a part of the non-cyclic photophosphorylation. We have the synthesis of ATP here between cytochrome F and cytochrome B6, and we have the synthesis of NADPH2 here after the formation of the ferredoxin as an important component of photosystem 1. Hence, the answer is D. Question number 23. In photosystem 1, the first electron acceptor is A, cytochrome, B, plasocyanin, C, an iron sulfur protein, D is ferredoxin. If you recollect, in the case of the light reactions of photosynthesis, in photosystem 1 from P700, electrons are accepted by initially an unknown compound, and the unknown compound is an iron sulfur compound. After it is taken by the iron sulfur compound, the iron sulfur protein, then it later on goes to ferredoxin. From photosystem 1, electrons move into an iron sulfur protein. The iron sulfur protein was initially not discovered, hence was designated as X. From X, it goes to ferredoxin, and from there we have the formation of NADPH2. So, among the choices in photosystem 1, the first electron acceptor, the initial electron acceptor is the iron sulfur protein, hence the answer is C. The most important word here is the first electron acceptor. What is this compound is the first initial electron acceptor, and later on at the second level it goes to ferredoxin. We go to question number 24. If thylakoids are removed and cultured in vitro, that is under artificial conditions, in a medium having carbon dioxide, water and exposed to light, hexose sugars are not formed due to A, carbon dioxide assimilation does not occur in light, B, light harvesting complex is absent, C, there is absence of carbon dioxide assimilating enzymes, and D, there is no linkage between P680 and P700. Imagine a situation wherein we isolate thylakoids from the chloroplast and we are culturing them under artificial conditions, which is termed as in vitro conditions. So, when you culture the thylakoids, in a medium and you are adding carbon dioxide, input is carbon dioxide, input is also water, as well as you also expose it to light. So, when you expose it to light, we find that hexose sugars are not formed. And the reason is carbon dioxide, the choice first one is carbon dioxide assimilation does not occur under light. and the second one is light harvesting complexes is absent. Third one, which is the most appropriate, says there is absence of carbon dioxide assimilating enzymes. So, when there are no carbon dioxide assimilating enzymes, that is when there are no dark reactions and when there are no biochemical reactions, or we do not have the most important basic enzyme like phosphoenol pyruvate carboxylase or RUDP carboxylase, then hexose sugars cannot get formed. So, in order that hexose sugars get formed in any sort of an experiment, there is a need for biochemical reactions. So, biochemical reactions are primarily determined by carbon dioxide assimilating enzymes. Hence, among the choices, answer C is the right one. Carbon dioxide assimilating enzymes, that is answer is C. We go to question number 25. Potassium hydroxide solution enables removal of dash from a controlled experiment during photosynthesis. It is A, C6H12O6, B is O2, C is CO2, D is H2O. Whenever in any normal classroom plant physiology experiment, whenever we use potassium hydroxide, we take a sort of a paper in which we apply potassium hydroxide and place it on a sort of a leaf. Then the leaf turns black, it does not turn green, primarily because potassium hydroxide takes and enables, it removes off the sort of a carbon dioxide. So, once carbon dioxide is removed, there is bleaching and a starch does not get formed. Hence, that patch wherein we have the potassium hydroxide, it remains pale. Hence, among the choices we have is the right answer is carbon dioxide, because carbon dioxide is removed by potassium hydroxide and it is termed as the sort of a moles experiment. The answer is C. Question number 26. Conversion of phosphoenol pyruvic acid to pyruvic acid and ADP to ATP are examples of A, substrate level phosphorylation, B, photoelectric phosphorylation, C, photophosphorylation, D, oxidative phosphorylation. Whenever we have phosphoenol pyruvic acid and it is converted to simple pyruvic acid, as well as when ADP is linked to this and it forms ATP, this sort of a reaction is termed as substrate level photophosphorylation. It indicates that there is a presence of a substrate, there is a formation of ATP in the presence of light, and there is a formation of a product. So, any chemical reaction in which there is an oxidation and a reduction, and again ADP is linked to inorganic phosphate to generate ATP, and ATP formation is linked with relation to the sort of an initial product and the final compound, and thereby the substrate changes, is termed as substrate level phosphorylation. So, the answer is A. Question number 27. During photosynthesis, one of the following can be termed as a first step: A, water ionization and splitting, B, joining of the 3-carbon compound to form glucose, C, excitement of an electron of the chlorophyll by a photon of light, D, formation of ATP and NADPH. Whenever we take photosynthesis, photosynthesis starts with the photon of light. When a photon of light hits a chlorophyll bed, the chlorophyll molecules get excited and lose an electron, and when they lose an electron, they have a charge. So, when they have a charge in the presence of a photon of light, so this forms the first step, or the light reactions are the first step in photosynthesis. So, among the choices, like water ionization and splitting is a secondary reaction, whereas predominantly to start light reactions we have two photosystems wherein we have a chlorophyll bed and a reaction center, and we have light getting dispersed and activating both the sort of reaction centers of the photosystems 1 and photosystems 2. Joining of the 3 carbon atom to form glucose is a part of the dark reactions, and formation of ATP and NADPH are again part of the end products of the light reactions. So, among the choices of A, B, C and D, choice number C, excitement of an electron of the chlorophyll by a photon of light, is the right answer. The answer is C. Question number 28. The flashlight experiment leading to enhancement of photosynthesis was studied by A. Hatch and Slack, B. Emerson and Arnold, C. Warburg, D. Benson and Kelvin. So, when you take the flashlight experiment, flashlight experiment in photosynthesis refers to studying photosynthesis by synergizing or combining a long wavelength of light with another short wavelength of light. So, when you give a short wavelength and a long wavelength together, that is together or simultaneously, when you flash a light, flash the long and the short wavelength of light on the chlorophyll beds or inside the sort of a chloroplast, we understand that the photosynthetic rate or the efficiency is greater. If you take red light and again you take far red light, in a flashlight it combines both red and far red light, and the efficiency rate, photosynthetic efficiency rate, is very much higher. So, because the photosynthetic rate is very much higher, it is termed as enhancement. So, this entire process has been studied by Emerson and it is termed as Emerson enhancement effect. So, the Emerson enhancement effect indicates how two wavelengths together can improve photosynthetic efficiency by a flashlight experiment. The answer is B. Question number 29. Which one occurs both during cyclic and non-cyclic modes of phosphorylation? A. Involvement of both PS1 and PS2. B. Formation of ATP. C. Release of oxygen. D. Formation of NADPH. When we take the light reactions of photosynthesis, we understand light reactions of photosynthesis has got both photosystem 1 P700 and photosystem 2 called P680. Now, what is common for both? Common for both is not just the involvement of both photosystem 1 and photosystem 2. In both, we have, whenever there is cyclic photophosphorylation, that is when it involves photolysis, PS1, quinone, electron transport systems, photosystem 1 as well as quinone and ferredoxin, as well as when it involves non-cyclic photophosphorylation, that is the Z scheme. The common point is they have the electron transport systems. So, because they have got the electron transport systems, there is always the formation of ATP. Whereas, release of oxygen is only predominantly allowed with relation to photolysis of water, which is specific for photosystem 1. Formation of NADP is specific for photosystem 2. Whereas, when you take ADP, ATP formation is common for both cyclic and non-cyclic photophosphorylation during photosynthesis. So, the common molecule is ADP, hence ADP is also termed as the sort of an energy currency of the cell, because it is going to be the sort of a source of power for the dark reactions to occur during photosynthesis? The right answer is B, that is ATP molecules. Question number 30. The fundamental features of Cran's anatomy in C4 plant is A. Chloroplast in epidermal cells, B. Rudimentary chloroplast in bundle sheath, C. Chloroplast in vascular tissue, D. Well developed chloroplast in bundle sheath. When you take Cran's anatomy, Cran's anatomy is a unique feature of C4 plants. The meaning of the term crans is wreath, W-R-E-A-T-H. It means a garland, and it has been studied by a German scientist whose name is Kranz. So we have the formation of a wreath-like structure around the vascular bundles. When you take the vascular bundles in C4 plants, we got the xylem and the phloem. The xylem and the phloem is surrounded by a specialized tissue which shows Cran's anatomy. So in Cran's anatomy, we have got the bundle sheath cells. This is surrounded by the mesophyll cells. If we take the sort of a leaf anatomy. Now, when you take the bundle sheath cells, here if you take the chloroplasts, the chloroplasts are large, more in number, they have high starch grains, but they are agranal, so they have only stroma. So because they have got only stroma, they are capable of very efficient dark reactions. And again, if you take the bundle sheath cells, in the bundle sheath cells we have the C3 pathway, and during the C3 pathway there is the presence of an enzyme called ribulose diphosphate carboxylase. So presence of more starch means higher photosynthetic efficiency. So what are the basic features of the Cran's anatomy? In Cran's anatomy, there is internal carbon dioxide fixation, and in time as well as also more importantly in space, that is photosynthesis occurs in mesophyll cells separately, spatially separated, and also in bundle sheath cells also it occurs. So thereby there is spatial separation of photosynthetates in the case of the C4, in the case of primarily in the case of C4 plants. So C4 plants have got Cran's anatomy. Whereas when compared to CAM plants, in CAM plants there is temporal separation of photosynthetates. When you say temporal separation of photosynthesis, in cross-solution acid metabolism plants, the photosynthetates get formed in time during the night and during the day. Whereas in C4 plants, the photosynthetates stay in the bundle sheath cells as well as in the mesophyll cells. This specialization is due to the presence of the Cran's anatomy or the wreath anatomy, which is a special feature of tropical plants which shows C4 pathway for higher photosynthetic efficiency. So among the choices, we have right choice is D, which states that well developed chloroplasts in bundle sheaths.